Hey, good afternoon, everyone. I'm Jishnu. I'm an assistant professor at the University of Pittsburgh here. I'm a computational systems immunologist. I run a research lab of around 20 people. And the fundamental theme of our lab is to infer mechanisms of immune regulation and dysregulation using machine learning and networks. And we use multi scale, multimodal data sets, genomic, cellular, molecular as inputs, use machine learning and networks to infer mechanisms of immune regulation and dysregulation. So, two broad themes, and you just heard Alyssa from my lab present how we think about networks, protein protein interaction networks, as well as gene regulatory networks to integrate genomic, epigenomic, and transcriptomic data sets to uncover molecular phenotypes of immunological disorders. I'll talk more about the machine learning aspects of what we've been working on, specifically a new interpretable machine learning approach that we developed for multi-omic integration. But before I get into it, I keep using this term interpretable machine learning. So, what exactly am I talking about? Is it just jargon, or are we talking about something biologically more interesting? So, as we all know, deep learning has revolutionized prediction. But in there are two kinds of machine learning that I think about. One is focused primarily on the prediction piece, where we are only interested in making accurate predictions, finding biomarkers or correlates. The other is the inference piece, where we want to move beyond biomarkers or correlates, beyond the what to the how and the why. And I reason that in human systems immunology, we need inference beyond prediction. So, how do you put together these high dimensional multi-omic data sets and move beyond just predictive biomarkers or correlates to inferring actual pathophysiological mechanisms? And to do this, we developed SLIDE, significant latent factor interaction discovery and exploration. It follows a lot of work. This is something we published earlier this year in Nature Methods. I'll only focus on what we did with Slide. We always acknowledge people at the end of the talks. I would like to do it at the beginning because we run out of time at the end. This was in collaboration with colleagues Alok, Bob, and Amanda, and co-led by two really good graduate students in my group, Javad and Hanshi. So what SLIDE does, it's a model to identify not just biomarkers or correlates, but to try and infer from these high dimensional multi-omic data sets, move to inference beyond prediction. And how we do it is actually very simple. So whatever we can measure using current technology is the observable features. Those are the X's. Most methods move from the observable features, the X's, to the Y's, which is the outcome or group of interest. We said, well, obviously, whatever you can measure is a limitation of current technology. You have single cell RNA, attack, proteomic, metabolomic, lipidomic. But biology doesn't care about what you can measure. Biology cares about the true underlying mechanism. So there are these latent factors or underlying latent states. And we want to move from the observable features first to the latent factors, and then move from these Z's, these hidden states, to the outcomes or responses of interest. Now, obviously, we did not come up with latent factors. Latent factors have been around in the statistical literature for 2 decades or more. So, what's unique about what we did? The first thing is we came up with a latent factor discovery approach with identifiability guarantees. So most methods out there, non-negative matrix factorization methods, do not have identifiability guarantees, meaning every time you do the non-negative matrix factorization, you get a different answer. So people run algorithms like consensus NMF to get an average solution. On the other hand, to get to identifiability, there are methods such as principal components regression where you have orthogonality, but that is not biologically real. Can we now have overlapping Z's that are not orthogonal but yet identifiable? So, working with theoretical statisticians at Cornell, we solve this as a constrained optimization problem where the only assumption we make is that each latent factor is anchored by two pure variables, meaning two features belong to that latent factor and no other latent factor. Everything else can be shared or not shared in a data dependent manner. So we have this latent factor discovery approach with identifiability guarantees without assuming orthogonality. Uh, Now, because we have this, we can regress using these latent factors. If they are non-identifiable, regressing using these latent factors does not make any sense. But because we had these latent factors that are identifiable and non-orthogonal, we can regress using these in a two-step fashion. The first is the linear component, where we go from these z's to y's using a linear model. But remember, these z's are multicollinear, meaning if we just use standard regression coefficients, the p-values could be deflated. So we used a modern method for false discovery rate control developed by Emmanuel Candace and colleagues at Stanford called Gaussian knockoffs, which allows us to control the overall false discovery rate. So using these identifiable latent factors, we now first identify the significant standalone latent factors. And then using hierarchical OR logic, we use a nonlinear model where we look at interaction terms and again use knockoffs to identify significant interaction terms. Slide has a lot of unique properties that distinguish it from other latent factor methods, competing methods, including MOFA plus, variational autoencoders as implemented vanilla VAEs, or even SCVI. I will go into actual concrete examples that illustrate this better. So the first data set that we applied this to was a single-cell RNA-seq data set in conjunction with Bob Lafiatis's lab to identify what we call putative causal latent factors of scleroderma severity. So this is standard single-cell RNA-seq data obtained from punch biopsies. So this is people with systemic sclerosis. Their skin is thick. They do punch biopsies at 18 different points in the skin and score it on a scale from 0 to 3 and 18 different points in the skin. So 0 means completely healthy, 3 means the thickest, the most disease. So the dynamic range, this is the outcome variable known as the modified Rodnan skin score, is a clinical surrogate of disease severity. And we use these transcriptomic profiles to identify, infer what could be underlying the pathogenesis of scleroderma severity. So, but before we get to the inference piece, we also looked at the prediction piece. So this is model performance. So slide and essential regression are the two methods that we've developed in a k-fold cross-validation framework with permutation testing. We do as well as state-of-the-art methods, including lasso, VAEs, MOFA plus, or FATE, coupled to regression, principal components regression, PLSR. But what's more interesting, I mean, the prediction piece is kind of boring. The goal is not really prediction. The goal is inference. What new biology have we actually learned? So this is a busy slide. These are the latent factors. So we have thousands of transcript abundances. We generated overlapping clusters of latent factors, hundreds of latent factors, and we have narrowed it down to these 10 latent factors that we can use to infer the underlying pathophysiology of systemic sclerosis. This is a busy slide, so I'll break it down into 3 parts. Each box is a latent factor. Anything with a green boundary is a significant standalone latent factor. Anything with a purple boundary is an interaction term. Now, the transcripts, the color represents the cell type of origin, as in the legend. And there's a dotted line in the middle of the slide. Anything on the left is associated with higher disease severity. Anything on the right is associated with lower disease severity. There were a few things that we found which make perfect sense, which people in the field knew. So, for example, we saw CCL19, IGF-BP5, and so on in these profibrotic fibroblasts, the myofibroblasts, which Bob and others know to be causal in pathogenesis. We also saw crosstalk between myeloid inflammatory and interferon signaling. This again agrees with what people know in the literature in terms of myeloid cells and fibroblasts, and these altered myofibroblasts being drivers of disease pathogenesis. But what was new in terms of what we found? 2 things. The first was we saw, in addition to myeloid cells and fibroblasts, and keep in mind that we are recapitulating this known biology without any prior knowledge. So slide is recapitulating this known biology without any prior knowledge. We found this intriguing role for keratinocytes, and this was an ignored cell type. But we've since gone back and done validation experiments. We really think that there is keratinocyte-centric pathogenesis mechanisms previously unelucidated that this method was able to converge on. We also see an intriguing role for HLA-DRB5. Obviously, HLA is super important in autoimmune disease, not as well appreciated in scleroderma, but the latent factors converge on that. So now I said, well, we capture linear and nonlinear relationships. Is that true? When we look at the genes within these latent factors, we see that we do capture both linear and nonlinear relationships. On the left, I'm also showing you what these interaction terms capture. So the blue is the empirical null when you just have a size match set of random latent factors. Green is the more interesting thing, where you actually see You keep the standalone latent factors the same, but you size match and shuffle the interaction term. So you can assess systematically compared to an empirical null as well as a partial null whether your model is capturing more information. Now, I keep using the word inference. What exactly are we getting? I don't use the word causality because when I put my statistical hat on, and we've done work with Takis Benes showing that this is actually linked to causality, but when I put my biological hat on, without a perturbation experiment, you cannot demonstrate causality. So I would say infer putative mechanisms of causality, and this is one instance of a human perturbation experiment that we did. So these were scleroderma patients treated with tofacitinib, which is a pan-JAK inhibitor, and we had their MRSS scores before and after treatment. And what we recapitulated was the known mechanism of action. So the difference, so the responders versus non-responders, was stratified using this IL-6 JAK-STAT-centric latent factor that we captured de novo, showing that we are recapitulating a known mechanism of action. We have also applied this to spatial transcriptomic data sets. This is with Amanda Poholik's lab, where we can capture, I won't go into details on that, in a murine model of allergic asthma, partitioning of immune cells in a lymph node. We get both the prediction as well as the inference piece. We've applied it to 10x Visium, slide seq data. But now I'll move on to another interesting biological example where, in conjunction with Alok Joglika's lab, we asked this question: can we identify latent factors underlying a cellular phenotype? So in scleroderma, we were using an organismal phenotype, disease severity, but now we are looking at a cellular phenotype, extent of clonal expansion of these CD4 T cells in a non-obese diabetic model of type 1 diabetes. Alyssa also presented some results from that in a different setting. And we have paired single-cell RNA-seq data and TCR-seq data. Each cell is labeled as unexpanded, single, low clonal expansion, or medium to high clonal expansion. And we asked, what are the latent factors underlying the extent of clonal expansion of these CD4 T cells? Of course, we can do prediction very, very well, but that's kind of the boring piece, as well as state-of-the-art methods are better than them. But the inference piece is, again, really what's most interesting. We found these 4 latent factors, these context-specific groups that we infer. Some of the genes in these are well-known markers like LAG3, PD1, activation markers, inhibitory receptors, or on the other hand, we have things like L-selectin or CCR7 markers of naive CD4 T cells. But what we rationalize is that the group structure matters. So in terms of these overall groups outperforming, and we compare to, again, other latent factor approaches, including MOFA plus or SCVI, the effect size per latent factor is much higher. SCVI, there are a lot of issues. It's obviously a variational autoencoder. It's non-identifiable, so the latent factors are unstable. We capture better effect sizes. We also find things, if we look at differential expression analysis, a lot of things people ask, well, you could just have done a DE analysis, so what would you have missed? What we would have missed is some of the genes in these latent factors pass univariate significance thresholds, others do not. But it is the context-specific group and this multivariate model that gives us what we statistically, with this kind of FDR control, think necessary and sufficient to infer what is underlying the phenotypes of interest. It could be an organismal phenotype or a cellular phenotype. And when we now look at well-known markers like LAG3, TIGIT, PD1, CD200, of course, higher in the expanded cells, cell and CCR7, higher in the unexpanded, but it's not a golden bullet. On the other hand, the effect sizes for these latent factors show a much cleaner trend. So in summary, SLIDE can integrate high-dimensional multi-omic datasets with no assumptions regarding data-generating mechanisms to identify significant latent factors underlying outcomes. These are putative causal, and some inferred mechanisms have been validated using perturbation systems or model organisms, comes with rigorous guarantees regarding identifiability and inference, FDR control, and the discovery of nonlinear hierarchical relationships. And using SLIDE, we've identified these in a range of contexts. The paper is out in Nature Methods. The code is also on GitHub. Please feel free to take a look. And of course, none of this would be possible without my lab, my awesome lab of around 20 people, a dynamic group of people from different backgrounds. And I would like to finally thank my funding sources. Without any of these funding sources, this work wouldn't have been possible. And thank you, and happy to take any questions.
That was a great talk. I just wanted to get some clarification on the mention of identifiability. So I know that we're putting on different hats. When we compare SLIDE to like things like SCBI, when we talk about the latent space being identifiable, I think with respect to things like SCBI, identifiable should mean slide is identifiable and is the with respect to the parameter estimation.
Yes.
Like you can pinpoint the parameter.
Yes. Yeah, that is correct. And the identifiability of slide, that's a great point. Thank you for clarifying, is with respect to the parameter space. So we have tuning hyperparameters. Given a fixed set of hyperparameters, we have one unique decomposition because of the way we have posed this constrained optimization problem.
That was a great talk, thank you. Despite the fast pace, I was able to follow everything, and I feel like I really understand everything. That's fantastic. So, what I'm kind of curious about is like when you were showing us the slide, the latent space variables, the green and the purple ones. Um, what I've done, so when you have a machine learning model, I guess just those you would start from latent space variable 1, 2, 3, 4, 5, and so on. So how do you go from latent space variable 1 to a particular cell type?
So that's a great question. So the framework is generalizable. So as I discussed, we have the observed features and we decompose them into latent factors, overlapping clusters, and then we are using regression to find the linear and the nonlinear, the significant standalone and the interaction terms. The way, so whether you know the cell type of origin depends on the data set and how we featureize the original X matrix. So in the context of scleroderma, for example, where you had the cell types of interest, we had around 15 different cell types, and we picked the 100 to 200 highest variance genes for each of the cell types. So the X matrix, the input X matrix, is the number of samples across all cell types, and we have just stacked the features across. So you're capturing both cell intrinsic and cell extrinsic. And the interesting part is this decomposition is done using the covariance matrix. So the SFRP2 cluster or latent factor, without prior knowledge, found only things within that single myofibroblast-like population. But there were other latent factors that had multiple cell types. But it could also be in the context of the CD4 T cells, we only have one cell type, the CD4 T cell. So it could be used for both cell intrinsic and cell extrinsic circuits.
I see. So basically, the way you were able to identify the cell type is because that particular latent variable was only linked to a specific subset of the features, and those features all correlated or were implying to be that particular cell type.
Exactly. Yes, and that is a feature of the result. That's specific. That's yes. Yes.
All right, let's thank our speaker again.